Good morning. So this group of awards that we're going to announce today are some of the most prestigious awards, if not the most prestigious awards in biomaterials. And as the chair of the committee, we were really struck by the incredibly strong candidates and the number of candidates that are members of our society and biomaterials community that are very strong candidates for this award. So one of the things that I was just having a short conversation with someone and said, you know, it truly is an incredible honor to be selected for these awards because of the other strong candidates that were nominated. So for the first award, this is the Clemson Award for Basic Research, and this was announced at the opening ceremonies, and we weren't able to give the award to the individual because of conflicts of time. So we'd like to do that this morning. This award is the Clemson Award for Basic Research. This is given to someone who has contributed to the basic knowledge and understanding of the interaction of materials with tissue. And this year, this award is being given to Philip Messersmith from Northwestern University. Philip?
Good morning. Thank you for all attending. My name is Kevin Healy. I'm a professor at UC Berkeley, but I was also a professor with Phil at Northwestern for some period of time. And I'll give you a bit of advice, Phil, that Monty gave me, which is be careful. That little circle falls off in about a week. So make sure you glue it back on. Keep that well placed in your office. Okay. So it's my true pleasure to do this introduction and welcome Phil into the family of Clemson awardees. Phil has had a distinguished career, as many of you know. He started out as an undergraduate at University of Illinois. He graduated in 1985. He then received a master's from Clemson University in 87. In 1993, he received his PhD under Sam Stoop in organoceramics. And I think, Phil, one of the things you mentioned to me was that these materials were some of the most problematic and hardest materials to work with, even though they delivered a lot of nice publications for you. In 94, from 92 to 94, he was a postdoc in Cornell University with Professor Gianellis working on silicon nanocomposites. And then in 94, he also started his first position at University of Illinois Chicago. So the theme is Phil likes Illinois, I guess, right? We met in about 1997. Largely, I was the chair of a search committee at Northwestern University in the dental school. And Phil was the top candidate chosen for that search. So myself and Professor Jeremy Gilbert, who were faculty at the time at Northwestern in the dental school, feel vindicated in our decision because of his distinguished career and also the close friendships we developed over the years. Although it was a rocky start for Phil, in 1998, that's a year after he started, the dental school was announced it was going to close. So there we were, the three of us, Jeremy, myself, and Phil, deciding what we wanted to do with our careers. So Jeremy was maybe the smartest at the time. He took off and went to Syracuse University. I stayed around with Phil, and we were reappointed in another department in the medical school. And all through this, Phil is, of course, trying to start his young career, and his lab hasn't been renovated, and he's sort of struggling and figuring out how he's going to manage all this. So he just buried in and wrote a couple of grants, and he got all of them, really, a number of NIH awards, including a first award in 1997, and this continued a series of awards, a Merit Award in '06. Continuing along the theme, he was a visiting professor in Japanese JSPS fellowship, and he's also done a significant amount of conference organization, including organizing this conference in 2010, the GRC Conference in Biointerface Sciences in 2010, and in 2010 was also elected as a Fellow of the Royal Society of Chemistry. He's also a Fellow of AMBI. He was elected in '06. And to get back to the story, he eventually moved up to the Evanston campus as a professor in bioengineering, and that appointment was made in about 2000. Since then, he has, his early work at Northwestern was focused on liposomes filled with calcium, as these calcium-triggered materials would basically calcify in the presence of a bath of phosphate or phosphate-containing buffers. These were early ideas of using in situ forming materials, which later became more important as he discovered, as we say, DOPA, right? And so DOPA is the molecule he'll tell you a little bit about, catechols in particular, which has a fragment of the DOPA molecule. And it was really 2003 where a seminal JACS paper came out from his lab focusing on DOPA and the use of DOPA as an adhesive. And from there, the use of DOPA has become, I think, a core part of his lab. I'm going to stop in a moment, let Phil come up here and give his presentation. But I'll just tell you, he's served on over a number of NIH panels. He started a company based on this technology. He's also given over 200 lectures and has published in some of the most illustrious journals, Science, Nature, Langmuir, JACS, and so forth. So with that, I'd like to personally and give Phil a heartfelt congratulations on this deserved award. Thank you.
Thanks very much, Kevin. That was a very generous introduction. So, as Kevin mentioned, we have for about 12 or 13 years been studying muscle-inspired biomaterials. Let's see, is this a laser pointer? Oops. I guess not. There we go. Okay. And that's going to be the subject of my talk. So if your only experience with mussels is as a menu item in a restaurant, which is what this picture is representing, my hope is that I would convince you by the end of this talk that mussels should be an item on the biomaterials menu. If you look at this picture, there's nothing that's immediately apparent in terms of how it adheres to surfaces. So let me show you a picture of that. This is the mussel byssus on the right. This is the attachment structure of the mussel, and there's only one function for this tissue, and it's totally cell-free tissue, which is mechanical adhesion. And the mussel secretes each one of these threads and adhesive pads in a very interesting process that, if you're familiar with polymer processing, is similar to injection molding. And so that's an interesting process for us, and also interesting with respect to potential inspiration for medical adhesives, because most surgical adhesives have have that property of beginning as a liquid, ending as a solid. And so that's one of the inspirations that I initially found in this unusual place. The other feature that's important to mention is mussels are famous for attaching to virtually all surfaces, and they do so under fully submerged and wet conditions, which is not a insignificant accomplishment. And then finally, over the years, a colleague of mine, Herb Wade at UCSB, has determined that the proteins that are found in these tissues are very unusual. And so my talk will focus on some of the components in these proteins and focus in particular in this area here where the adhesive pad is secreted and attaches to the surface. And so those are very unusual proteins. And as Kevin mentioned, one of the defining features of these proteins is this amino acid DOPA. It's derived from tyrosine. So the proteins are secreted and post-translationally modified to add this second hydroxyl group. And this side chain is referred to as a catechol functional group. And DOPA is really found throughout that entire abyssal structure, including the threads as well as the adhesive pad. Most notably in the proteins that are found at the interface. And those proteins can contain upwards of 25% of DOPA, which is, and DOPA is not incorporated in proteins in high concentrations in any other organisms except for a couple of marine organisms that utilize adhesives. And it's not found in vertebrates, vertebrate proteins anyway. And so when we started this work in the late 90s, it was generally recognized that DOPA is important in several aspects of this adhesive structure. One, of course, is adhesion. And this was driven by the observation that the DOPA content is highest at the interface. And also in cohesion. Cohesion, that meaning, to me, meaning the elastic properties, the bulk properties of this tissue. DOPA is very important. But few of the details of that, of surrounding DOPA and its roles were really understood on a molecular level. And so it's very gratifying to me to be given this award and the Clemson Award for Basic Research, because we invest a lot of time in trying to understand what's going on in this tissue. And so a little bit of that I will tell you about this morning. One of the methods that we employ is single molecule force spectroscopy, a very oversimplified cartoon of a very complicated experiment is shown lower left here, which is essentially, it's an AFM experiment, not imaging, but rather a force experiment. So we derivatize an AFM cantilever with PEG, which, as you know, would render this cantilever tip probably very poorly interacting with surfaces. And then we attach a single molecule terminal group of one of these chains. And this is one of the challenges of the technique, is being able to say that you actually do have a single molecule. But we can do this, and then we do a simple experiment, simple in principle, which is we put the cantilever down on the surface and then pull away, and we want to know how much force to take that molecule off the surface. And this is some example force curves. These are essentially the raw pieces of data. The feature that we're interested in is in this area right here. These are it's fairly small, but these are fairly large peaks as single molecule force spectroscopy goes. And you see actually some of these force curves are labeled or shown in red, others in blue, and that's because you can see that the ones in red have a very higher pull-off force than the ones in blue. And we can do this experiment many times. Under certain conditions, we could do it many times. We could take the cantilever off, put it back down, and that's exactly how we generate this data that you see here, which is all obtained from a single cantilever, okay? Which means that we can generate force histograms. And this, from this experiment emerges one of the interesting features of DOPA, which is the equilibrium that exists between DOPA and its quinone form. And that's shown up here. This is an equilibrium that is shifted to the right under alkaline pH. Marine pH is 8.5 usually. And so the quinone in these proteins, as they're secreted to form the abyssal tissue, there should be actually both of these residues present. So if you do the experiment I just described, generate the force histograms, you can see these force histograms here. The one shown in red, which is the higher force histogram, is due to the interaction of the catechol with the surface. This experiment is on TiO2. And the quinone, as you can see, has a much lower pull-off force. Okay. Now, what about tyrosine? As I mentioned, tyrosine is derived, or DOPA is derived from tyrosine. So if you do the same experiment with tyrosine, you find a very low pull-off force. So this immediately tells us that the addition of the second hydroxyl group to form the catechol is very important and enhances dramatically the adhesion with the surface. Now, on TiO2 and most other inorganic surfaces, you might conclude from this data that the quinone is relatively uninteresting. Where the quinone gets interesting is when you have surfaces that are organic in nature. And of course, the muscle, when it encounters a surface, it's often a surface that, even if it's a rock, it's pre-fouled with macromolecules, bacteria, algae, etc. So there are certainly organic molecules, and some may be primary amines. And under these situations, actually the quinone forms a covalent bond with primary amines. And then when we do this experiment, we pull on the cantilever, we break a covalent bond. Okay? And these are the very few observations that you see out here in the 1 to 3 nanonewtons range. That's the range at which covalent bonds rupture under applied force. Okay? And so this is a tremendously versatile chemical landscape, a force landscape that's derived from a versatile amino acid that has a very complicated chemistry that allows us to interact with an inorganic surface and an organic surface throughout a range of types of interactions and a range of forces. And this is probably very important for the muscle. As I said, it encounters very heterogeneous chemistry when it attaches to surfaces. Now, these numbers sound very small, and they are small. But if you're familiar with force landscapes of chemical bonds, you would know that down here near the origin, below 10 piconewtons, is the force necessary to rupture a hydrogen bond. Most receptor-ligand interactions in biological systems, like avidin, biotin, integrin with its ECM ligand, rupture in this area here. So this type of interaction in the 600 to 800 piconewton range for a single amino acid on an inorganic surface is truly an exceptional adhesion bond. The second role for DOPA is in cross-linking. DOPA provides essentially the cross-linking chemistry to drive the liquid to solid transformation in the adhesive. Remember that BISA structure is secreted from liquid protein precursors, and in about 3 minutes it solidifies. And the chemistry underlying all that is due to the presence of DOPA. And this is the equilibrium I already described with the quinone. And when the quinone forms, it will cross-link with other DOPA molecules to form protein crosslinks, and this gives rise to a liquid to solid transformation. This type of crosslink was found probably about 15 years ago in the actual muscle abyssal tissue. The pathway at that time was really not well understood, and so it is probably still debated. The number of possibilities, like enzymatic oxidation to drive this oxidation, or redox. And here's where metals come in to the story. It turns out that the muscle byssus is highly enriched in metals. I've listed two of them here, the two of the more important ones for our research, which is iron and zinc. In both cases, the byssal tissue has about 5 orders of magnitude higher metal concentration than the surrounding seawater. And it turns out that the muscle mobilizes these metals and co-injects them with the proteins during formation of the byssus. And so there's a lot of interesting questions about what is the mechanical role of the iron. Why is it there? There's nothing diffractable in terms of mineral content in this tissue. Okay? And here's where more recently in our group as well as other groups, we've been able to show that actually through redox reactions, iron is involved in this oxidation, which gives rise to covalent cross-linking. And then iron is also involved, and this happens under a secretion pH, which is acidic. And iron is also involved in the formation of mechanically active coordination bonds that look like this. They're tris coordination bonds between one iron atom and 3 catechals. And this is favored at marine pH, this type of coordination between iron and catechals. This catechol functional group is very well known in the literature for its strong affinity for transition metals. And this interaction here is mechanically active. And this has led us to recently propose a rather complicated formalism for the role of iron and catechol in the system, which is essentially gives rise to a cross-linked protein matrix that's cross-linked both by covalent bonds as well as these coordination bonds. And under applied force, these coordination bonds are actually labile. They're sacrificial in a sense. They can be ruptured under applied force and then reform in the absence of load. And this we showed in actually a synthetic polymer system. This is where actually a synthetic biomaterial can actually help us understand the biological system. This is an 8-arm PEG molecule with the end groups are all catechals. We formed a hydrogel by mixing with iron and then equilibrated that gel to marine pH and studied the bulk mechanical properties. And just to make this story very short, the hysteresis you see here between loading and unloading curves is due to the rupture and then reformation of these sacrificial iron catechol coordination bonds, and it actually significantly toughens this material. So this is why we think that these coordination bonds are sacrificial. They dissipate energy when they rupture, and so they toughen the material. Then more recently, we've looked at the role of zinc as well, and this involves a different residue, histidine residues in the core collagen. So the threads are made of modified collagens. Very interesting proteins as well. They have N-terminal and C-terminal regions which are incredibly enriched in histidine. Almost every other amino acid in those regions is histidine. And they're also enriched in zinc. And so this hypothesis of zinc being also sort of forming a sacrificial mechanical bond has been around for a while as well. And this is something that we're actually currently looking into, again, with single molecule force spectroscopy by probing the force of rupture of this bond between 2 histidine residues and bridged by a zinc. And you can see some of the preliminary data here that we have, which gives us bond ruptures in the 400 piconewtons range, which again is very, very large for a single molecule observation. So these are likely also the interactions between zinc and histidine in this tissue are also likely to be very mechanically active and contributing to the mechanical properties that we observe of the tissue. And one of the things that has driven this hypothesis in our study is that if you pull on these threads, you can pull them past their plastic deformation limit, and then they self-heal over a period of a few hours to recover their elastic properties. So it's a very interesting system, and we believe that it's the sacrificial rupture and then reformation of these coordination bonds that gives rise to the recovery, self-healing, if you want to call it that, of the tissue. So those are examples of our truly fundamental research, which is aimed at trying to understand this interesting tissue. So now with the rest of my time, what I'd like to do is give you a couple glimpses of how we use that information. And the idea here is that we embrace is to learn about the biological tissue and use that to inform the design of synthetic materials. And the examples I'll give you are in the area of medical sealants and adhesives and a few examples in coatings. So to do this, we do a fair amount of polymer synthesis. This is just meant to be a general description of some of the polymers. The architectures might be branched, they might be linear, and the idea is I think I've already shown you in a couple of the structures, is that we derivatize end groups or side chains with catechol, possibly also other amino acids that are found in the muscle adhesive proteins. Here's an example lower left of one of these polymers. The one that I'll focus most of my remaining talk on with respect to medical adhesives. And it's a fairly simple construct. Not fairly simple, it's very simple. It's a branched polyethylene glycol with catechol end groups. Not even a dopa on the end group, it's just the catechol. Oops. And I would point out also that the concentration of the catechol in this polymer is much, much lower than in most of the muscle adhesive proteins. However, in the presence of the catechol in this polymer, we have all the chemistry, as I've described, to do two important things with medical adhesion. One is to drive a solidification reaction from a liquid precursor, as a liquid medical sealant, to a solid, and do that rapidly. And then the other chemistry, as I've described as well, is that once oxidized, these can interact very strongly, covalently, non-covalently, with tissues, inorganic tissues as well as organic tissues. Okay? And this polymer can be formulated into a medical sealant using a conventional two-barrel syringe device, polymer in one, oxidant in another. They mix in a chamber here, come out as a liquid and gel in a few seconds or a minute. So we've explored in a number of contexts the medical applications of these gels. I don't have time to show you a video of vascular sealing, but this is one of the earliest applications that we explored. Antibacterial hydrogels, actually, if you use as the oxidant to drive the oxidation of the catechol to the quinone, if you use silver ions, the silver gets reduced to form nanoparticles. Silver oxidizes the catechol. You get the cross-linking and adhesion. And embedded within the gel are silver nanoparticles, and these are very good for antibacterial properties. We've also used these sealants for islet transplantation. Here the idea is to simplify transplantation itself through the use of a sealant which can be used to bond the tissues without cutting or suturing of the tissue directly onto the tissue surface. So here you can see in this image here, at the top is the tissue, the islet here, and shown below is the adhesive. So the adhesive is simply used to bond the islet onto the tissue. The islet gets revascularized from the tissue underneath. And the one I want to spend a couple minutes on is this one here, fetal surgery, which we've become interested in just the last few years. This is actually there's not a lot of biomaterials people working in this space, but there's a great need for materials for fetal surgery. Fetal surgery has only been around since 1980. Mike Harrison at UCSF did the first open fetal surgical procedure in 1980. And it's done for a variety of reasons. Some of them you can see here. For example, it's done for correction of spina bifida defect. It's a very challenging discipline. And one of the reasons it's so challenging is that Of course, to do any procedure, either endoscopically or open surgery, you have to cut through the fetal membranes, okay? And these membranes really do not heal very well. And many of these procedures the surgeons want to do at a very critical time in development, usually in the weeks 20 to 35, where survival of the infant, if it's born prematurely, is quite compromised. And so there's significant challenges. So premature rupture of membranes, or PROM, is what we're talking about here. This can be either a surgically created rupture or a spontaneous rupture, and it gives rise to risk of infection, premature labor, as I mentioned. PROM is present as a contributor to just a few percent of births, but represents over 30% of neonatal morbidity. And it's been called, because of this, it's been called the Achilles heel of this surgical field. So to make a long story short, we're exploring our sealants, the ones that I described already for sealing fetal membranes. Here's a mechanical device used, and draped over this device is a human fetal membrane. So this is fresh discarded tissue. And we use this device to stretch the tissue, and then we poke a hole in the middle with a fetoscopic trocar. It's about 3.5 millimeters, so not a huge defect, but not a small one either. And then we simply apply the liquid from that applicator I showed the picture of onto the tissue, let it solidify, and then examine the strength of that bond, as well as if you cut through and do histology through the middle of this defect, you can see the result that's shown below, which is the fetal membranes here in pink in the middle. The darker materials are muscle mimetic adhesive, shows very good adhesion to the tissue, and importantly also bridges this defect here. We can do, because this is fresh tissue, we can do about 24 or 48 hours of organ culture and look at the very, very acute stages of inflammatory response through general histology here, and compare it to some other candidate materials. The two on the right are cyanoacrylates, Dermabond and histoacryl, and you can see a very significant disruption of the tissue here. So it's a very aggressive acute inflammatory response with these materials. The two in the middle here are other versions of polyethylene glycol sealants. They don't seal or adhere to the tissue very well, as you can see. This one actually cannot even be found bound to the tissue after 24 hours. And this is our muscle mimetic material, as I mentioned. Fibrin also adheres well to the tissue, but it's dissolved very rapidly. It's not suitable for fetal membrane sealing. We're currently at this stage here with a rabbit fetal study. This is actually an interesting study. It's given us some glimpses of hope that this could be a very useful biomaterial. If you expose the fetal membranes and poke the hole but do nothing to fill it, you get a survival rate of about 36%. If you fill it with our muscle glue, the survival rate rises to 60%. If you use the muscle glue plus decellularized amnion, we get about 80% survival. And fibrin glue plus the decellularized tissue remains fairly low. So we're pretty hopeful about the outcome of this, and are planning actually sheep in vivo studies later this year. So in my last few minutes, I just want to talk a little bit about our work in coatings. And here, well, if you believe what I've told you so far, you would perhaps predict that catechol, DOPA, and other catechol are very good adhesive chemistries. And so we can take advantage of that to graft molecules, biomolecules, polymer molecules onto surfaces for a variety of reasons. And so we developed, starting in 2003, a family of surface modification chemistries that it might involve, for example, a catechol derivatized polymer or a catechol initiator, which bonds to the surface, and then we can therefore functionalize surfaces using catechol as an adhesive chemistry, maybe in the same way that you might use a silane on a metal oxide, or an alkane thiol on a gold surface, except these are tremendously more versatile than those other chemistries. Those other chemistries are very, very precisely tuned to one type of substrate. And the catechol works on most substrates, actually. So one of these I will mention, which is inspired by one of the muscle adhesive proteins, MEFP5, which has the highest DOPA content, but also has the highest lysine content. So this is, you can think of this as a polycatecholamine polymer. Okay, and so we developed a surface modification strategy that derives from a small molecule. You can think of this dopamine molecule, which has the amine derived from the lysine, the catechol from the DOPA, as a monomer. You think of this as a monomer, and it's a very simple surface modification strategy. You take a solid, it might be solid bulk material, it might be fibrous, might be nanoparticles, microparticles, immerse them in a solution of dopamine at 8.5, and out comes after a few hours a conformally coated solid. And you can see some of the pictures here that indicate this. And this works on pretty much any type of material using the same basic chemistry. And so in our first publication in this area, we showed that then you can take that and think of it as a primer onto which you can then add other functional properties. You can graft polymers on the surface to make anti-fouling surfaces, or you can make them cell adhesive. This is an example of binding HA onto surfaces for cell attachment. So I have about 1 minute left here, and I just want to leave you with this thought, which is that we can sometimes even go places where evolution and nature really have never had a reason to go. And this is one example. I described for you in some detail here the muscle adhesive strategy, which is permanent adhesion, unusual proteins, takes advantage of strong chemical interactions, and works under wet environments. At the other end of the spectrum of biological adhesion is the gecko, and the gecko relies on temporary adhesion using ordinary proteins and weak interactions, and only works under dry environments. And the adhesive mechanism relies on intimate contact of little tiny pads on the foot of the gecko. And so we can, oops, I don't know what that did. So we can actually combine these two strategies into an interesting material that has both the features of the muscle adhesion, wet adhesion, with the gecko. And this is actually a tape-like adhesive material. You can see the short stubby pillars here, and on top of which we coat a polymer that's derived from the muscle adhesive protein. And this material shows a 15-fold increase in wet adhesion and is a temporary adhesive that works both in dry and wet environments. So I am out of time. I'm going to summarize by saying that we're inspired by muscle adhesion as a source of inspiration for biomaterials, particularly medical sealants and coatings. This applied interest is supported by a lot of basic research. I have a lot of people to thank, including the people that really do the work. My current and former students is a picture of my current group, and I hope I haven't left anybody out. These are the people that are really phenomenal and have made this research all possible. It's also possible, of course, because of funding. I've been quite fortunate to have a lot of continuous funding from NIDCR and NIBIB and some other sources that you see here. Also a lot of phenomenal collaborators. There are a couple of people I really like to thank, though. Herb Waite, who is the person that has devoted his entire career, 40 years, to studying muscle adhesive proteins. I wouldn't be standing here if he hadn't discovered these proteins, and so I owe a lot to him, and we've become great friends as well. And then Jeremy, Gene Lautenschlager, and Kevin. Kevin described already the situation that arose at Northwestern in my first couple of years, and these guys were really important in keeping me sane during a pretty insane period. and then, of course, my family. Most of all, my wife Sherry, our kids Alex and Kaylin. All three of them sacrificed a lot to allow me to play with mollusks. So with that, I will thank you as well for your attention.